10. Okay. So here, this one is a repeat slide ban siya. Illustration showing contributions of mesoderm and neural crest cells to the formation of the skull. Wala? So neural crest cells, this one, the blue structures, neural crest cells contribute to all phases of the bones of the face and part of the neurocranium. While your paraxial mesoderm, this one, the red one, the red structures, forms the part of the neurocranium, while your lateral plate mesoderm, dili siya sa laryngeals. Okay, so muscles are derived from paraxial mesoderm, with the exception of the pupillary muscles, remember, which came from your ectoderm. Okay. Okay, this one showing the cartilaginous derivatives of each arch. The specific cartilages derived from each arch are indicated. No? In addition, arches contribute to bony structures not directly derived from the cartilage. And this one is the summary of the innervation and derivatives of the pharyngeal arches. So we have arch 1, 2, 3, 4, and 6. 4 and 6. Okay. Hindi ko gigawang matukso. Hindi ko mudlay man sini. Hindi kang sabat kulbaan ko. We proceed na lang. Okay? So, this is an illustration showing the pattern of breast cell migration and their HOX gene expression. Okay? So, there are 3 major paths of breast cell migration. Cells originating from the hindbrain, okay, from this region, migrate from the segmental regions called the rhombomeres. Note that the rhombomeres 3 and 5, this one, ang white, do not contribute cells, which are patterned by HOX genes, this one, and crest cells carry this pattern of expression with them into the pharyngeal arches, where the expression will contribute to regulating the differentiation of each arch. So crest cells from the midbrain, here, region express OTX2, which contains the homeodomain but is not a member of HOX gene family. Okay, this one, pharyngeal pouch gene expression regulates patterning of the pharyngeal arches. Then we go to the the growth of the second arch that includes the remaining clefts. One of the first only the first cleft, no? Di ba external ang cleft sa sulod pouches. So, only the first cleft makes the contribution forming the external auditory meatus and the ectodermal covering of the eardrum. Okay? So, ang first cleft mo will develop into ears na siya later on. No? Naging auditory meatus siya. The remaining clefts are obliterated. Okay? Wala siya nag-appear. The remaining clefts are obliterated by caudal proliferation and growth of the second arch that merges with another proliferation of tissues called the epicardial ridge. Okay? And sometimes this process leaves remnants of these clefts behind as a cyst or fistula. Nag-form siya cyst later on kung nara pa siya. So these are the location of cervical cysts and fistula. Wala siya. This one are, kung naman siya sa liog these are the lateral cervical cysts and fistula, the locations. Sometimes may kabukol pa diri sa tlunggan, no? And this is the region of preauricular fistula. May buho siya. Amo na siya. So dira nag-originate ang mga abnormalities during the formation of the pharyngeal clefts. Okay. So next, we go to the derivatives of the pharyngeal pouch endoderm. Dito naman sa sulod. And then, endoderm of the pouches likes to proliferate and differentiate into glands in the neck. No? So endoderm forms the functioning cells of these glands, while mesenchyme forms the connective tissue. So the first pouch, dito naman sa sulod, expand to form the tympanic cavities and auditory or eustachian tubes. Okay? Tympanic cavities. The second pouch naman, this one, form the palatine tonsils. Okay? Sa throat tanan niya nga part, no? The third pouch, this is the tympanic, no? The palatine. Okay, the third pouch, and the fourth, this one, the fourth pouch makes the parathyroid glands. But contrary to what might seem to be appropriate, the third pouch makes the inferior parathyroid, and the fourth pouch makes the superior parathyroid. Okay, so the third pouch makes the thymus that migrates to the thorax. The fourth pouch naman niya makes the ultimobranchial brachy that attach to the thyroid gland. The thyroid gland forms the proliferation of endoderm from the floor of the pharynx and is not a pouch derivative. Okay? Thyroid gland. Okay, let's move on. We go to the thyroid gland formation and migration. So thyroid gland tissue forms in the midline of the floor of the pharynx. This proliferation forms a thyroglossal gland or duct that opens into the pharyngeal opens into the pharynx as at the foramen cecum. Okay? And glandular tissue forms at the tip of the duct and migrates to the neck anterior to the laryngotracheal rings. So this is our thyroid gland. In front of the tracheal rings, no? Muning atong trachea. So this slide shows us the sites for ectopic thyroid tissue and thyroglossal cysts. Okay? So normally, the thyroglossal cyst duct, the thyroglossal duct, and foramen cecum degenerate. Katula na siya dapat. However, sometimes cysts form from these regions that fail to close. No, nag-open siya. Ectopic thyroid tissue can also remain anywhere along the migratory path of the gland. So cysts and ectopic thyroid tissue are always located in the midline. Sa midline siya. Okay, that's all for the thyroid. We go to tongue development. So tongue develops on the floor of the pharynx from the lingual swellings. No? So these swellings are formed by the proliferation of endoderm and underlying mesenchyme. So initially, the swellings form from the first 4 arches. Okay? But the third soon overgrows the second. The third soon overgrows the second, and thus the body of the tongue, or the anterior 2/3, is derived from the first arch, while the root derived from the third. Okay, so the origin of the tongue is reflected in its sensory innervation. The anterior 2/3 being innervated by the mandibular branch of cranial nerve 5, the nerve to the first arch, no? May summary yata, wait. Okay, wala pa, later. Tongue muscles are derived from occipital somites that are innervated by hypoglossal nerve or cranial nerve 12. Okay. Basta mo ng tsura sa tongue during development. We go to the formation of facial prominences. So here we have the electron micrograph, no, scanning micrograph showing formation of this one, the facial prominences. So at 4 weeks, the pharyngeal arches are visible laterally, okay, and the facial prominences can be seen when the embryo is viewed from the frontal direction. So at this stage, the fronto-nasal prominence and the 2 maxillary prominences are apparent, as is the mandibular arch. So 2 thickenings of ectoderm, the nasal placode, this one, ng Arionto, may depression siya. Nasal placodes begin also to form on the lateral surfaces of the frontonasal prominences. Okay? Ang manitsura yan. So this one, illustration showing continued growth of facial prominences. So endoderm in the center of the nasal placodes proliferates and invaginates to form the nasal pit or the primitive nares. Primitive nares nato na siya, ilong, no? Primitive ilong. This invagination together with proliferation of the surrounding mesenchyme forms the nasal process, medial and lateral on each side of the nasal pits. So the maxillary prominence continue to enlarge and grow anteriorly. Okay? And the mandibular prominence, this one, merge across the midline to form the mandible. So, unlike other facial clefts, no, mandibular clefts are very rare. It seems that the mandible is almost completely formed, no, as soon as the mandibular portion of the second arch appears. Sometimes a depression or dimple forms but not a cleft. There is a tunga. Okay? Sometimes may mga cleft ka sa tunga. Abnormal, kagali. Di ba? wala lang nag-close properly ang mandibular prominence mo. O, di ba? Okay, this one. Okay, we go to fusion of facial prominences to form the face. Okay? This is a photograph of a human embryo showing contributions of facial prominences to the formation of the face. So, 2 medial nasal prominences. Okay? This is our lateral prominences. Medial nasal prominences merge, no, to form the tip of the nose. So, fuse this to form the tip of the nose, the philtrum region, this one, okay, of the upper lip, and the primary palate, okay, and the maxillary here. The maxillary prominence also fuse with the medial nasal prominences here to complete the formation of the upper lip and alveolar ridge, no? So the lateral prominences, this one, okay, this one. The lateral prominences form at the sides of the nose. So, ari siya, no? Sides ang may buo na. So, the frontonasal prominence forms the bridge of the nose. Okay? Dali na lang ma-form ang inyong itsura at 10 weeks of development. So still on the primary palate formation, in addition to the forming of the tip of the nose and philtrum, the medial nasal prominences form the intermaxillary segment. There is a sulod, no? If pangangahon mo patient, a baby. The intermaxillary segment is composed of the labial component that forms the philtrum, an upper jaw component that includes the 4 incisor, okay, 4 incisors, incisor teeth, no, and palatal component that forms the primary palate. Okay. Okay, so here we have the maxillary prominences and the secondary palate formation. In addition to forming of maxilla, no, by fusion of the medial nasal prominences, the maxillary prominences extend into the oral cavity and will form the secondary palate. Dito sa likod sa primary palate. Okay. So here on our next slide, you can see that there's a formation of the secondary palate and the palatal shelves. Okay, this one. The palatal shelves form from the maxillary prominences. Okay, dito rin. Extend into the oral cavity, and these shelves assume the vertical position on each side, on either side of the tongue. Okay. So this illustration of the secondary palate development and the palatal shelves here fill up, no, to assume the horizontal position dorsal to the tongue, and for this movement to occur, the tongue must drop out of the way. This one, manaog siya dapat. And in order to the tongue to move ventrally, the mandible should grow forward to create a necessary space. Okay. So this one is again the illustration of the secondary palate formation at 10 weeks. Once the palatal shelves have elevated, they grow and fuse across the midline. Okay. And forming the secondary palate and the soft palate and the uvula. Okay, the uvula na siya. So fusion occurs by mechanisms similar to those utilized in the neural tube closure. So clefts occurs when fusion process fails or when the shelves fail to elevate. So we have the line of fusion. Okay, these are the line of fusion. So ma-fuse sa tunga, ma-fuse sa kilid, ma-fuse sa kilid. Okay, so the primary and secondary palates fuse, and at the midpoint of these lines of fusion is the incisive foramen. Okay. So we have here the picture of a 7-week embryo showing where facial clefts occurs. Okay, so indentation in the fusing and merging regions of the facial prominences at 7 weeks show clearly where different types of facial clefts can occur. Okay, ato ba lang wala nag-move, wala siya nag-fuse. Okay, those are the origins of your facial clefts. So here we can see the clinical pictures of children with different types of facial clefts. So cleft lip, no? Cleft lip is more common than isolated cleft palate. Okay, cleft lip more common. So cleft lip is more common in males, 80% males sila, while yung cleft palate is more common in females. Okay, ang lip sa male, females palate. cleft lip is more common in most cases are multifactorial. Lain-lain na siya. So, anticonvulsants such as diphenylhydantoin, ano ba yan? Diphenylhydantoin or phenobarbital have a propensity for causing cleft lip and palate. Okay? So, this is your lateral cleft lip. There is kilid lang siya yan. One side. This bilateral, no? Cleft lip. This one, lip and palate git, no? Ang problem nato ni is to feed, no? The baby. Okay, this one, only the palate. Nami siya sa babaw. This one, oblique facial cleft. Di ba? Na sila dapat confuse before. So, this one, wala siya na-confuse. Okay? So, we have here the picture of a child with midline cleft lip. Midline cleft lip. So, tunga siya. So, midline clefts arise due to failure of the medial nasal processes to merge across the midline. So, these clefts are more serious than the lateral clefts because they are often associated with varying degrees of brain defects and mental retardation. Okay? So, another picture of midline type of defects. So sa amo ni siya ang normal siya, no? So during time of development. And then this is median cleft lip. Kaluoy sa ila. So we go to the illustration showing the when midline is established. So midline is established during the third weeks, no, of development, including eye field, which are initially a single field across the midline. So SHH, no, gyapon, is the gene that is critical for establishing the midline, including separating the eye fields, no. So note that the midline is established approximately 3 weeks, a time when most moms do not know that they are pregnant. So because alcohol, among other things like retinoids, can cause holoprosencephaly, it is easy to see how someone who says she will stop drinking when she gets pregnant may not have stopped, no, prior to this critical period. Another example why birth defect prevention strategies must be implemented before conception. See? Nam-inom, chill. Okay? So, this one illustrates showing the pathways of neural crest cell migration to the face. So, recall, no? The neural crest cells make up of most of the bones of the face. So, also, for no unknown reasons, they are very sensitive to population of cells that can be adversely affected by genetic and environmental insults. Okay? So here we have defects due to the neural crest migration. So we have Treacher-Collins syndrome. A maxillary process are targeted, see, resulting in deficiency in the maxilla and zygomatic arches. Which causes the eyes, no, to appear to be bulging. The ears are also severely malformed. In Robin sequence, mandible is deficient. Dili wala siya mandible. And so there is no room for tongue to drop. Okay? And allow the palatal shelves to flip up and fuse. So consequently, these children have cleft palate, micronatia, and a posteriorly placed tongue. So any of these neural crest cases can also have heart defects, no? Remember? Because crest cells help to septate the heart. The craniofacial and cardiac malformations occur together. Okay? And last slide, these are clinical pictures of the children with craniofacial defects. We have the George anomaly. A part of the spectrum of defects that includes velocardio facial and conotruncal anomalies, no? Due to deletions in chromosome 22q. Okay? Deletions of 22q. Ang di George, most severe, no? Because in addition to facial defects such as hypertolerism, microstomia, small mouth na siya, no? And the patient's lack of thymus gland and suffer immunological problems. Okay. How about your oculo-auriculovertebral spectrum? Golden Haar, no? Tawag na sa ilang golden Haar syndrome or hemifacial microsomia, characterized by defects in the maxilla, temporal, sa mga kilid naman siya yan. And zygomatic bones, ear defects, wala siya ear, eye tumors, vertebral anomalies. Okay? So for some reasons, these defects are usually unilateral, one-sided siya. So all of these different neural crest defects can be caused by retinoids, no, alcohol, maternal diabetes, and other teratogens that may affect crest cell migration and or proliferation or cause a cell death in crest cells. No? Tunto na kay crest cells ngayon. Okay, so this is my last slide for the second lecture. Break ka ta. Break the night. Okay lang, let's have, what time is it? 3:42 pa lang ah. Okay, so we go back at 4 o'clock. Ha? Hingi ka mo, anay?
Take it noted, Doc.
Yes,
Thank you.
Doc.
Okay.
Thank you, Doc.